Hey Team Bio, welcome to your very brief screencast on gene regulation. Now, we've been talking extensively about the central dogma of biology, this idea that DNA is transcribed into RNA and leads to protein, which leads to traits and behaviors. But some of you have asked really excellent questions about the fact that while all of our cells, every cell in our body, with the exception of our gametes, have the same DNA, we express really different proteins depending on the cell type that we're looking at, which leads to different traits and behaviors within cells that contain the exact same DNA. So how is this accomplished? And it's accomplished through a process called gene regulation. This helps control what's called cell differentiation, the fact that your neurons are very different from your muscles, and it also helps with energy efficiency. So, we're making sure through gene regulation that the neurons are expressing the neuron proteins and the muscles are expressing the muscle proteins. And we're also, through gene regulation, only expressing the proteins that we need when we need them. Okay, so this is how cells that all have the same DNA can have very different phenotypes, traits, and behaviors, and it's through how the genes are expressed. Okay, so know that this example that I'm going to give you is a very simple example in bacteria, and gene regulation in eukaryotic systems is much more complex, but starting out with a simple example allows us to extrapolate to more complicated examples later. Okay, so what we're looking at here is what's called the lac operon. And the lac operon is the system of genes that controls the digestion of lactose. Oops. What's going on here? Oh, this is huge. Okay. Controls the digestion of lactose. Many of you know lactose. It's a milk sugar, or it's often found in milk. It is found in milk, among other things. And it's composed of, it's a disaccharide, so two sugar groups together. It's glucose and galactose. Now, people who are lactose intolerant lack the genes to break lactose down into glucose and galactose. Because lactose enters their gut undigested, there are bacteria in there that anaerobically break down the lactose into glucose and galactose for them and creates all sorts of cramping and gas as a result. Okay, so this is how bacteria turn on the genes that are responsible for the digestion of lactose to break it down into its component pieces, glucose and galactose. Okay, so here's how this system works. Now, if there's no lactose in the system, it doesn't make sense to create the genes that are going to break lactose down into its component pieces because we don't have lactose to digest. So the way this system is set up is to prevent the transcription and then translation of the proteins that digest lactose when lactose is absent from the system. So there's a regulatory gene that is always on. We call this constitutively on. The regulatory gene codes for an mRNA, which codes for a protein that is a repressor protein. The repressor protein can bind to the DNA, and it can block RNA polymerase from binding to the promoter and moving down the operator into the lactose utilization gene area. So when the repressor is bound to the DNA, these genes can't be transcribed by RNA polymerase, and therefore we can't make these proteins. But this makes sense. If there's no lactose, why would we create the genes or the proteins that break lactose down? So it's more efficient to produce no lactose or lactose utilization genes when no lactose is present in the system. Okay, now, so this is the operator turned off because lactose is absent. Now let's examine the situation where lactose is present. Lactose is a substrate of the repressor gene. And when lactose binds to the repressor protein, it changes shape. Now, the protein, this repressor protein, can no longer bind to the DNA. It doesn't fit anymore because it's bound lactose, its shape has been changed, can't bind to the DNA. This means that RNA polymerase is free to bind to the promoter region, and it slides on down the gene system and it transcribes the mRNAs, and then the enzymes, the proteins for lactose utilization, are going to be created. Now, in bacterial systems, it is not uncommon for one mRNA to contain chunks of multiple proteins. This is not true of eukaryotic systems. One mRNA copy is going to lead to one protein product. But in bacteria, several proteins can be stacked one after the other on one mRNA transcript. Anyway, that's a bit of a segue. So these are the enzymes that are going to break down lactose and convert it to glucose and galactose. Okay, so let's examine the system. When these genes are created, when these genes are transcribed and the proteins are translated, what happens to lactose levels? Well, we now have the proteins to break lactose down. So lactose levels, which are initially high, begin to fall. And lactose levels continue to fall because we have the proteins, the enzymes, that are chopping lactose up into glucose and galactose until we get to the situation where lactose is gone. It's all been digested. Oops. It is glucose and galactose. Okay, so now we are in a position where, once again, lactose is absent. If lactose is not present in the system, then the repressor protein no longer is holding it, and it is once again free to bind to the operator region. When it's bound to the operator, RNA polymerase is prevented from attaching to the DNA, and the lactose utilization genes are no longer transcribed. So the enzymes for lactose utilization, their levels now fall. Which is good for the bacteria because it doesn't want to waste energy making these proteins when lactose is now all been digested. So this is called a negative feedback loop, which I have a little image of here. So a negative feedback loop is where if you have an initial effect A. So this, let's say this is the presence of lactose. This is going to cause a change in property 1, which is the transcription of those genes. And the effect B is going to be that now glucose is now the genes are present, I guess. And now this is going to start digesting the lactose. And so now the lactose levels fall again. So this is a dampening loop. And this is a very basic example of how genes are regulated. Okay, that's all.